the last big topic, application, but like mixing everything together. So 2 more to go here. Okay. So the part 1, application of biotechnology. So first of all, we can make use of this biotechnology to produce different type of protein again. So what can some example protein for pest resistance may be toxic for the pests, even to give the new protein to bacteria so that they can remove some toxins, some oils, leakage. Some protein is medicine, is just blah blah blah, something like that. So make sure you know. But if we focus on mammal proteins, then it will more on the health part, okay, the health issue. Of course, the most common example will be the insulin, okay. So if you want to use insulin as example, you should be able to tell the importance, how we can produce them by using bacteria, E. coli, how we collect them, purify them, put it into medical use, something like that. So just some example if we want to make use of recombinant bacteria to make protein for human. Okay. Let me see. So we can make insulin, growth hormone, we can increase food production. So these are just some side information here. Okay, so we can make insulin, human insulin, using non-human cell, not even animal, using bacteria cell. We can also make some growth hormone. So just some example of the application of these techniques. We can also increase the food production by making GM food. Of course, mainly on plant, but we can also increase the food production in animal. And we can also make use of the recombinant technique to make vaccine. So vaccine is something like traditional now. So you know how the traditional vaccine work, but we also have something called the recombinant vaccine, vector vaccine, subunit vaccine, DNA vaccine. So these are something more recombinant. So make sure you know about this new type of vaccine. For example, DNA vaccine, what are they? They are some DNA directly injected into our body. They are of course genetically engineered, okay, so that they can trigger some immune response, but not too strong to kill you. Okay, and because you design the DNA so well, so they will act exactly the same like the pathogen. So your body can respond to it, triggering the immune response, and the next time you are really infected by the pathogen, you can fight very well. Okay, so these are different type of vaccine using the techniques of recombinant technology. And of course, we have gene therapy. Okay, so we also have some presentation last week talking about gene therapy, so you should be more familiar with it now. So know about gene therapy, mainly about how they can treat the genetic disease. Of course, you have to first know about what is genetic disease. And if it is a genetic disease, then it is possible to be treated with gene therapy. For example, if there is a gene in your body that is mutated and therefore become not functional or causing disease, then you can use gene therapy to replace it. Or if there is a gene mutated, you want to turn it off. Okay, you are not replacing it with a copy, but just inactivate it so that they won't express themselves to produce some protein that's called the disease. Or you can introduce a new gene. Maybe sometimes the gene is not mutated, or they are missing at all. You lack some gene in your genome, and therefore you can try to put a new gene, bring the gene back into your body to fight the disease. Okay, so some possibility of using gene therapy, and then the procedure of how to carry out the gene therapy, how does it work. And we also have to think about how to deliver the gene into our body. We can have viral delivery, we can have long viral delivery. So long viral is something like the transformation actually. So how to put the DNA into our body. And viral delivery is something a bit different because we have to make use of the virus, and there are different choices for us. to pick from. Different type of virus, they have their different advantage, disadvantage, but we can make use of them to bring the DNA into our body. Okay. So yeah, some example of how to treat some genetic disease with gene therapy. And then we have PCR again. Okay, make sure you know raw material, major steps in the PCR, blah blah blah. So more details on the major step. Denaturation, breaking the DNA apart, annealing, sticking the primer to the DNA, and lastly, elongation, making the DNA longer and longer. So make sure you know the step, you know the material involved, temperature, sometimes you have to raise it, sometimes you have to cool it down, and the final product, something like that. Okay, PCR, and some more on PCR, different type of PCR. So these are more like practical thing. Okay, you can have some brief idea. We have, and then sequencing. So we always have to check the genome, check the gene. Okay, so PCR is just amplifying the DNA, but sometimes you want to know really exactly what DNA is there, then we do sequencing, and we talk about some different type of sequencing, the single sequencing, shotgun, next generation sequencing. Okay, so have some basic idea on how they work, why you can tell the sequence by doing so. Okay, we are also able to synthesize short fragment, not too long, short, but enough to be used short fragment of DNA. And how the gel electrophoresis work. Okay, make sure you collect it white, the white charge, otherwise the DNA will just disappear from your gel. Okay, and then lastly, the last